My name's Eric Kaler. It's my great honor and privilege to serve as the president of Case Western Reserve University. And one of the very special parts of my job is being part of ceremonies like the one we're going to have this afternoon. We're going to celebrate the appointment of Dr. Evan Daenerys as the inaugural John Seibyl, MD, Professor in Neurodegenerative Research. We're also going to celebrate the appointment of Dr. Jacqueline Zosneski. How did I do on that? I practiced all afternoon, so that's good. Zosneski. As the inaugural Catherine Seibyl, BSN, MSN, MPH, Professor in Nursing, Research, and Caregiving. I think your business card will have to be 3 by 5 to get all of that on there, but that's okay. Congratulations to you both, and welcome also to your family and friends who are able to join us today. I'd like to begin by acknowledging several special guests who are here this afternoon. First is Bob Pavey and his wife, Trish. Bob is a member of the university's Board of Trustees. Welcome. We're also joined by the Dean of the School of Medicine and Senior Vice President for Medical Affairs, Stan Gerson, who you'll hear from in a minute. And you'll also hear from Carol Musil, Dean of the Francis Payne Bolton School of Nursing, who's here today. And of course, we welcome John and Kathy Seibyl, as well as their family and friends. Now, as you all know, John and Kathy are both distinguished alums of Case Western Reserve University. John graduated from the School of Medicine in 1986, and Kathy first earned her undergraduate nursing degree here in 1977, and then her master's in nursing from the Francis Payne Bolton School of Nursing in 1981. Here's a fun fact. Just a week after John earned his degree, the couple were married at the Amasa Stone Chapel. Case Western Reserve is not often thought of as a super romantic place, but it turns out it can be. So after that, John went on to join Yale University's School of Medicine, where he eventually served as chief of the section of nuclear medicine. He later co-founded a nonprofit called the Institute for Neurodegenerative Disorders, followed by the company Molecular Neuroimaging, LLC, now in vitro. John describes himself as a clinical scientist, entrepreneur, Parkinson's patient advocate, and most importantly, a long-suffering Guardians and Browns fan. And brother, I feel your pain. It's really, oh my goodness. Yesterday, don't get me started. So he's semi-retired now, at least he is on paper. I chatted with him before the ceremony, and he freely admits to being pretty much failing retirement, but with practice, maybe it'll get better. We'll see. Kathy worked as a pediatric nurse practitioner and conducted epidemiological research for the U.S. Department of Veterans Affairs. Today, she spends much of her time educating John on what it means to be retired, while also being a fiber artist and a weaver. Importantly for us today, both John and Kathy are philanthropists and humanitarians. In 2021, the couple made a transformative $7.5 million gift to their alma mater to support multidisciplinary research in neurodegenerative disease at their respective schools. The gift was intended to enhance the university's ability to create ripple effects, those are Kathy's words, to benefit patient outcomes in Alzheimer's disease, Parkinson's disease, and multiple sclerosis. And one portion of their gift established a fund for neurodegeneration innovation in research, care, and education at both schools. This fund supports multidisciplinary research and training in neurodegenerative disease to improve the care for those whose lives have been impacted by neurodegenerative disease, as well as the lives of their families. Another portion supports fellowships at both schools to train the next generation of research scientists in brain health. And the final portion of the gift established these two inaugural professorships that we celebrate on this stage today. So John and Kathy, we literally cannot thank you enough. So many of us know someone whose lives have been impacted by this disease or loved ones who have dealt with this in their family. We're so grateful for your support, so grateful. And at this time, please join me in welcoming John and Kathy to the podium to share a few words. Please.
Well, thank you for that lovely introduction. And good afternoon, everyone, and thank you for joining us today for this celebration. We especially want to thank our family for being here, some who have traveled great distances to join us this afternoon. John and I want to extend our congratulations to both Evan, Daenerys, and Jacqueline Sosnowski in their respective endowed professorships at the schools of medicine and nursing. We also want to thank Deans Carol Musell and Stan Gerson, as well as Pamela Davis, former Dean of the School of Medicine, as persons who initiated this process of discernment as to what our gift might become and the dimensions it might take. Along those lines, we are very grateful for our relationships with members of the Health Science Development Office. In particular, Mike Dulcic, I don't know where he is, but it was over dinner at our home in Maine that Mike helped us hatch the idea of what a gift might look like. Today, John and I are filled with gratitude for the education we received here at Case that has helped us in ways we have never imagined and has continued to help us grow. The former political analyst of PBS NewsHour, Mark Shields, once shared a valued wisdom he learned from his parents and treasured his entire life, which is, every one of us has been warmed by fires we did not build, and every one of us has drunk from wells we did not dig. We couldn't agree more, and with this bit of wisdom that enlightens us today as we celebrate these inaugural professorships. So let us all find a moment to be grateful for the warmth and drink we have been gifted with by others. And now I'm going to let John, where are you, speak to why we made our gift, why to Case Western Reserve University, and what it means to us on a more personal level.
Thank you, Kath. As I mentioned to some people, I'm a little better at cerebration than celebration, but we'll do our best here. First, I want to echo Cassie's thanks and share her sense of gratitude to the leadership of the Schools of Nursing and Medicine and the development group for the collegiality and the teamwork in translating an in-code idea into some monetary resources into a multifaceted, multidisciplinary, unique research opportunities, which is characterized by the professorships that are given today. So many people have asked me, why did I make this gift? Why did we make this gift? And why did we make it to Case Western Reserve? For the first question, there are many reasons. Some are really very obvious. I have Parkinson's disease, and Kathy is a caregiver. Also, ironically, I'm a clinical research scientist who spent the last 3 decades developing imaging biomarkers to help diagnose disease, monitor progression, and even identify individuals at risk for Parkinson's, Alzheimer's, and other neurodegenerative diseases. Some other reasons we made the gifts are not so obvious and are embedded in our personal philosophy of the giving. It goes like this. All our lives we give and we receive. From the time we are infants, we are heated by the warmth of fires that we did not build, and later on we share the warmth of fires that we did build with others. Giving and receiving happen at many levels, the material, the emotional, the intellectual, and others. I like to think about this from using a pulmonary medicine metaphor and horticulture. So I have kind of a hybrid horticultural pulmonary model of giving. Giving and receiving are like inhalation and exhalation. The latter is a single process called respiration, upon which the vitality of the individual organism is dependent. Similarly, giving and receiving are components of one process, that of human engagement and interaction, the currency of our connectedness, and the wellspring and source of our vital humanity. Now on to the horticulture. Giving is also about planting seeds and letting them grow, perhaps being surprised at what results, what creativity is employed, what questions are answered, what new questions arise, and what positive impact can be made in the lives of the patients and their caregivers. For the second question, why CWRU? Again, there are many reasons. Suffice it to say that the combination of an impressive new facility designed for multidisciplinary interaction, the rich academic and faculty resources, and the history of educational entrepreneurism in health sciences suggest that the soil here is rich indeed. Hence, the decision we made to give to the schools of nursing and medicine was quite natural. You might even call it a no-brainer. Sorry about that. With all this in mind, let us take a deep breath together and celebrate, even as we rededicate ourselves to tackle the terrible problems of degenerative diseases. Armed with both joy and a sense of gratitude, may we plant the seeds that bear abundant fruit, and may our aspirations as to the particulars of that abundancy be the continued inspiration for the perspiration as we labor towards the harvest. Thank you.
Thank you. Thank you, John and Kathy. And we have for you a small token, literally a small token of our appreciation. They're not attached, so it takes one hand for this and one hand for your chair.
Okay.
The chair holders get bigger chairs later. And John, this one's for you.
Oh, thank you.
Enjoy those.
This is wonderful.
It's unusual. Thank you.
Try and sit on it.
No, don't do that. Very good. Now, please welcome to the stage our Dean of the School of Medicine and Senior Vice President for Medical Affairs, Stan Gerson. Stan, please.
It is indeed a pleasure to be with you this afternoon, and following on the comments of John and Kathy is going to be hard because of their incredible effort and dedication to the institution and to the causes that they've identified. On a lighter note, I've had the opportunity to present many endowed professorships, but if you'll pardon me, this is the first time we've done a mixed doubles. So both two schools and a male-female recipient. And it really does underlie the fundamental aspect of this dual professorship, and that is an effort to coordinate and support some of the basic science that might take place more preferentially in a school of medicine, and certainly the attention to caregiving, caregivers, and the consequences of disease in the individual undergoing that process, certainly in the space of a chronic disorder like neurodegeneration, that may take care on occur preferentially in schools of nursing. So this obligate connection, I think, is going to be actually important for decades to come for these two professorships. And so if there's a consequence you guys happen to get together and play pickleball, it's okay. It'd be within keeping. So we're proud of the Department of Neurosciences and the School of Medicine, of the effort that we undertake to understand neurodegenerative disorders. We had a really good interaction today with a couple of the projects that the Seibyls have helped us support that, again, bring the two schools together, and we're making slow but steady progress with, as I said, some spectacular scientists that are participating and collaborating in ways they wouldn't have without the instigation of the Seibyl family and couple and their involvement with us as we move these along. We're not going to solve neurodegenerative diseases quickly. We need to do far better at understanding their etiology, their background, whether it be using iPS cells, early detection, whether we're studying young children with a cognitive defect as their presentation of a neurologic disorder, young adults who've had brain trauma through their sports injuries, or older individuals who are suffering from the chronicity of neurodegeneration. And let me just point out that the professorship concept is such an important one for schools of medicine and academic enterprises. They certainly honor and recognize the accomplishments of our best and our brightest faculty, the contributions that they've made. They help us tether them here with that recognition and the support that they receive. We have over 125 endowed professorships in the School of Medicine, but we're always obliged to look for resources from generous donors, colleagues and alums who are able to help us provide the support for them over the long haul. And they really do help us with our overall mission of the School of Medicine, and you'll hear soon from the Dean of the School of Nursing, Carol Musel. For the School of Medicine, it's to improve global health by linking research to populations in a superb education environment, and we're certainly proud of that effort that we have ongoing here. Now, Kathy and John, you're following in the footsteps of endowed professorships at universities that began, we believe, in 1540, when King Henry VIII bestowed the first professorship at Oxford. The first one in the United States, we think, was in 1721, and at Case was in 1901. So you follow in a long tradition, and one that will survive all of us and lead to things that we cannot even imagine today. So now I'd like to introduce Dr. Evan Daenerys as the inaugural chairholder for the John Seibyl MD Professor of Neurodegenerative Research. Dr. Daenerys joined the faculty in 1990, is currently professor and interim chair of our Department of Neurosciences. He took on the interim chair position for a second time, so I thank you for that effort. Prior to coming to Cleveland, Dr. Daenerys earned his BS in chemistry at San Diego State, his PhD at UCLA, did postdoctoral research at the Salk Institute, where he started to focus on neuronal receptors, in the first instance, the nicotinic receptor, and he's now been working on the serotonin neurotransmitter, which is a pleiotropic transmitter important for almost every function of neuronal cells, which he will describe to us, and which the dysfunction of which is certainly associated with neuron and axonal disorders, with a variety of neurodegenerative disorders, and we're not quite sure sometimes whether it's the chicken or the egg in the process. So his current understanding of efforts is focused on the genetic mechanisms that act across the lifespan of the neuron and the serotonin system, and how its function modulates as we grow and develop, how it modulates with our external stimuli, and what the genetic changes that occur, which I'm sure he'll describe briefly, at least in English, that are linked to autism, intellectual disabilities, and his ability to then say these lay the groundwork for understanding future therapeutics that could be developed. I will say that as I asked him to take on the interim chair of the Department of Neurosciences, it was only because it was a unanimous decision on the part of his department that he said yes. So he is eagerly awaiting us to secure a full chair position so that he can get back to his research. And without further ado, I think Evan would like to tell us all about that. So Evan, please.
Well, that was a great job, Stan. You covered my research really well. So I'll just add a few extra details on that. It's truly an honor to be here and to be the recipient of the John Seibyl MD Professorship in Neurodegenerative Disorders. And I want to start out by extending my sincere gratitude to John and Kathy for this appointment. I'm truly honored. I also want to thank our former chair, Dr. Lynn May, who thought me worthy of being nominated for this appointment. And then I would like to thank Stan for recommending me to the Seibyls and to the Board of Trustees. So thank you all very much for this. I truly am grateful and appreciate it. So in the remaining minutes, what I thought I would do is I would describe my research path that led to my interest in neurodegenerative disorders. And so, it really all began during my postdoc work in La Jolla when I became interested in neurons. And through that effort, I discovered all of the beta subunits of neuronal nicotinic acetylcholine receptors. And that was through molecular cloning, and I didn't have any preconceived ideas that it was going to lead anything other than satisfying my curiosity. But it turns out that that work that I did led to the discovery of the major brain nicotine receptor in mammals, because that major brain nicotine receptor is assembled from alpha-4 beta-2 subunits. And that led to something that was something I didn't predict, that Pfizer would go on and develop varenicline, which is a partial alpha-4 beta-2 agonist for the alpha-2 beta-4 receptor. And that led Pfizer to develop and market a pretty darn effective smoking cessation drug called Chantix. So it's just a great example of how just doing curiosity-based research led to a really important translational event. So now this didn't really stimulate my interest or light my fire in drug design or development, but it got me interested in how complex patterns of gene expression, like this one that I discovered for beta-2 all those years ago, well, that one in the middle, all the brain sections, it got me interested in understanding how complex gene expression patterns are established in the brain to generate different kinds of specialized neurons. I wanted to know how these genes are regulated to establish different neuronal subtypes. And so in my lab at CWRU, my group went looking for regulatory factors that control neuronal identities. And what we found were two really important regulatory proteins called PET1 and LMX1B. And these two regulatory proteins, they're transcription factors, they are absolutely essential for building the brain serotonin system in the mammalian brain. And they do that in two ways. The first way they do it is by controlling all of the genes that encode all of the enzymes for converting dietary tryptophan into serotonin, thus enabling serotonin neurotransmission. And then what they do is they go on to control long distance axon outgrowth from serotonin neurons for expansive serotonergic neuromodulation. So serotonin axons invade almost every parts of the CNS, the brain, and the spinal cord. And because of that, they've been implicated in modulating just about everything we do physiologically and behaviorally. So we figured out the regulatory programs that make that happen during development, during embryogenesis, and the early postnatal stage of life. And so, it turns out that serotonin axons are really long. So are dopamine axons, so are noradrenergic axons, so are cholinergic axons. And there are some estimates out there in humans that some single serotonin neuron axons can be over 100 meters long. And I still find that hard to believe, but it was published by a reputable lab at Hopkins about 10 years ago. So I'm telling people that these axons that are diagrammed with those red arrows are really long because they not only send out a single axon branch, but a single axon path, but they also arborize profusely, like one of those trees out there that you see when you're driving up Fairmount Avenue. You got a main stalk and you've got all these branches. If you add them all up, it turns out in the human brain, they've been estimated to be over 100 meters long. And So, you know, long axons are really vulnerable to breakdown and to degeneration. And that raises some questions that, from our reading of the literature, have not been adequately addressed, despite their pretty substantial relevance to human health. And here are the questions that we think about a lot. How are synapses and axons, especially long ones, maintained for decades of life in mitotically quiescent neurons? All of our neurons were born before we were born. Most of them, the vast majority, are born before we were born. Their connectivity is made in our early few years of life. There's pruning that goes on later up into our 20s, but the connectivity is pretty much made when we're just young kids. And then it's got to last for life, for decades, in some cases over 100 years. How does that happen? Second, what explains the natural breakdown of synapses and axons that occur during normal aging? So as you age, axons are breaking down. Why is that? Third, why is this breakdown, this natural breakdown, accelerated in almost all neurodegenerative diseases? It seems that what goes first in a neurodegenerative disease is the axon before the cell body. Why is the axon so vulnerable? And our hypothesis is that axons, especially long axons and their synapses, have to be actively maintained in adulthood by regulatory proteins, transcription factor proteins that maintain these key genes for keeping axons going: synapse, axon, mitochondrial proteostasis genes, for simply homeostatic renewal of macromolecules. Macromolecules are turning over all the time. They've got to be resupplied in order to make that axon and synapse, the integrity of it, intact. And so our idea is that these programs are doing that in adulthood after development has happened. And in this way, they safeguard axons and synapses from degeneration for lifelong circuit function. And there are cases, you know, some people are living till 110 years old. They must have functional neurons that are being maintained through all those decades. So we wanted to test this. So we wanted to test the idea that there are these adult stage maintenance programs that are in the business of maintaining axons by maintaining those key genes. And so what we did is we took away from adult serotonin neurons these two key transcription factors. LMX1B and PET1 are expressed very strongly in adult serotonin. We took them away. And what they normally do is these two TFs, these two transcription factors, maintain hundreds of synapse, axon, and mitochondrial genes to establish axons and synapses. So when we took them away in adulthood, what we found was that these genes that they normally control, their levels plummeted. Hundreds of synapse, axons, and mitochondrial genes lost their expression. And that was associated with some pretty profound pathological morphological changes in the serotonin axons and their synapses. So the axons, for example, began to fragment into little pieces. That's what this diagram is showing, and we saw that under the microscope. And then what they developed are these swollen varicosities and these even bigger spheroids that were filled with pathological markers like amyloid precursor protein and alpha-synuclein. And the synapses, the synaptic boutons, started to retract from their cell bodies, and they began to fall apart and disappear. And so this indicated to us that, at least in the serotonin system, these long axons have to be actively maintained. Now, what's interesting gives an idea that what we're finding in serotonin neurons is of broad applicability is that LMX1B is also expressed in dopamine neurons, in adult dopamine neurons. And there's evidence that LMX1B levels are reduced in the surviving dopamine neurons in PD cases. And there's other evidence, not quite as comprehensive as ours, that LMX1B is needed to maintain synapses, dopaminergic synapses. So what we're finding in serotonin neurons may be broadly applicable through this connection between LMX1B and the DA system and the serotonin system for maintaining, safeguarding synapses and axons and from degeneration. So our working model is that the weakening of these adult stage maintenance programs, and this weakening could be brought about by intrinsic or environmental induced agents like DNA damage, double-strand breaks. This weakening leads to transcriptional decay and epigenetic noise for these key synapse axons, mitochondrial and proteostasis genes, that then in turn leads to the degeneration of synapses and axons and progressive neurodegenerative disease. That's our working model. And with the John Seibyl Professorship, my lab is going to continue to investigate this model to further explore the intrinsic mechanisms that safeguard synapses and axons against degeneration. Thank you.
Thank you, Dr. Daenerys. Very, very interesting work. Let me now welcome to the stage the Dean of the Francis Payne Bolton School of Nursing, Carol Musil. Carol.
Thank you, President Kaler, and thanks to all of you for joining us today to recognize and celebrate two remarkable women, Catherine Seibyl and Jacqueline Zazniewski. First, I want to acknowledge John and Kathy Seibyl, who make this day possible. We appreciate your groundbreaking vision to create opportunities for nurses, physicians, and others to come together to study brain health, neurodegenerative disease processes, and family caregiving in new ways that enlighten and transform our science. I also want to recognize Dr. Shirley Moore, former Associate Dean for Research at FPB, who helped make the initial contact between the School of Nursing and the Seibyls associated with her Brain Health Connection Center grant some years ago. And Shirley's joining us in the audience today. And now let's turn to Dr. Jacqueline Zazniewski. Jacqueline is a professor in the School of Nursing and a longtime colleague and friend. We have partnered together on many research projects focused on caregiving and interventions to support individuals dealing with acute and chronic physical and mental health problems, including dementia. The focus of her work for over 30 years is in the area of resourcefulness, cognitive and behavioral skills for managing stress. Her resourcefulness intervention has been tested worldwide and has been supported by NIH and foundation funding. She has mentored numerous doctoral students, junior faculty, writes prolifically, and has won countless awards. Because of her pioneering and extensive work with depressive cognitions, resourcefulness, and caregiving, Dr. Z, as she is known by her students, was the obvious choice to receive the honor of the Catherine Seibyl Professorship in Nursing, Research, and Caregiving. FPB is internationally recognized for its legacy of leadership and innovation. We now have 17 endowed professorships, the most of any nursing school in the country. When a professorship is certainly a prestigious honor for the faculty member as well as the school, the endowment is an enduring tribute and testament to the donors who support them. By funding endowed chairs, donors support the brightest minds and help them focus particular problems and issues, as well as spur advancements in their areas of science. This is especially true for Catherine Seibyl's professorship. As you heard earlier, Catherine is a double alum of FPB, having received both her baccalaureate and master's degrees in nursing here. She has a particular affinity for wellness and mental health. So today we come together to bestow the Catherine Seibyl Professorship in Nursing, Research, and Caregiving, and to recognize its namesake, Catherine Seibyl, and its first recipient, Dr. Jacqueline Zazniewski. Jacqueline, the floor is yours.
Thank you for that wonderful introduction, Dean Musil. President Kaler, Dr. and Mrs. Seibyl, and everyone here who has come to celebrate today two remarkable alums who have created this endowment, two alums from Case Western Reserve University. I am honored to be named the inaugural Catherine Seibyl Professor of Nursing, Research, and Caregiving. And I owe special gratitude to Mrs. Seibyl for this gift. Thank you. I also thank you for the opportunity to share a little bit about my NIH, my current clinical trial, that is examining self-management interventions in caregivers of persons with dementia. And these interventions are delivered according to their needs and their preferences. But before I get into that, I want to acknowledge my investigative team. I wouldn't be able to accomplish any of this research if I didn't have those people behind me that were supporting me and part of the investigative team. Dr. Christopher Brandt is my colleague and statistician from the School of Nursing, and University Distinguished Professor Eva Kahana is also my co-investigator. I'd also like to acknowledge my current supportive research team members. This list is not exhaustive. There have been many students and other persons who have made significant contributions to this particular research study, but these are my current team members, including my PhD students and staff members, project manager, and my intervention supervisor, Dr. Ivan Juratovic. Now, the short title of this particular grant is called the Caregiver Career Study. Truly, caring for someone with dementia involves a long-term commitment that might be marked by 3 distinctive stages that caregivers go through as they're providing care for their care recipient who has dementia. It starts with in-home caregiving, typically. And then there comes a time that perhaps the care recipient can no longer be cared for in the home and needs to transition to a care facility where they can get more care. But the family member is still very much involved, and they become a care partner in partnering with the facility to provide the care for their family member. And then there is the stage of caretaker, those caregivers that are bereaved when their care recipient passes away. Now, as family carers take on these new phases and transition in the dementia caregiving career, they face changes in needs, behaviors, symptoms, and the health of the person for whom they're providing care. And all of those things can adversely affect their own health, causes considerable stress, and can adversely affect their mental and their physical health. So the current study actually is examining interventions, strategies, ways that we can help these caregivers as they're going through their caregiving career to minimize their stress and to promote their optimal mental and physical health. So those of you that know me, I love my models. And so I'm going to present for you what is the just some highlights from the research. This is the theoretical model that we started with that involves actually three major conceptual areas: the context, the process, and the outcome. And the context I've already explained to you. It's the caregiving situation, whether they're the in-home caregiver or partnering with a facility, or whether they're actually bereaved. Now, the process involves the interventions. And we're testing in this particular study 2 interventions against an attention control condition. I'll tell you a little bit about the interventions as we move through this presentation. But these interventions have hypothesized effects on specific carer responses, and that is their levels of perceived stress, their levels of depressive thinking patterns, and their levels of negative emotions. And then ultimately, when we look at the health outcomes, these interventions are proposed to affect their health risks, their mental health, and their physical health. Now, in looking at the effects of those interventions on those carer responses and carer health, within the context of this study, we're also looking at whether it makes a difference what phase of the caregiving career the carer actually is in. So the study has at least 3 innovative components I'd like to highlight. First of all, it's the examination of the effects of interventions across the caregiving career, which is kind of unique for this study. Typically, research has examined caregivers in one of the phases, or maybe crossing over 2 of the phases, but to actually be able to put into the context of a single study the 3 phases of the caregiving career is a unique innovation. The second thing is the way that we are assigning for testing the effects of the interventions based on the caregiver's needs and preferences. Rather than just randomly assigning caregivers to 3 different intervention groups, we're basing it on their needs and preferences. I'm going to share with you how exactly we have determined that. And thirdly, the use of biofeedback as an intervention was particularly innovative, something that is rarely done in the caregiving literature, particularly in populations with dementia caregivers. So this is just a highlight of the design. We were targeting 300 carers across the 3 caregiving categories. And again, the first arrows you see, there's 3 arrows. The first says SM need, that's self-management need. We assigned the carers in that particular arm to get an intervention that is based on their need. And we determined their need by using validated measures, cut scores. And we gave them, for example, a resourcefulness scale to determine if they had low scores in resourcefulness, they had a high need for resourcefulness training. We also measured their heart rate variability, which is the gold standard indicator for our heart rate variability biofeedback intervention. So whichever one they scored the lowest on was their greatest need, and that's the intervention that they got. So the random assignment was based on need, not by intervention. Second group, the second arm, self-management by preference. We asked these carers, which intervention would you like to do? We have a cognitive behavioral program that teaches resourcefulness skills, and we have a biofeedback program. And we gave them whatever they preferred. That's the intervention that we gave them. The third arm was an attention control condition during which we taught them about different diversional activities that they might do. So our carers were recruited from the community, a variety of methods. That research team list that you saw worked very hard in posting flyers, newspaper ads, contacting community agencies, getting referrals, going through social media. There was just so many things that we used as recruitment methods for getting our carers. They were from Cuyahoga County and the neighboring counties. And then we needed to actually expand in order to increase our recruitment. We actually expanded another radius of counties beyond that. So you can imagine how many miles my team has put on their cars to go and do their data collection. Now, originally, I will tell you that the study was designed to use face-to-face data collection methods and face-to-face delivery of the interventions. Well, we had to transform most of the study, as much as we could, to online platforms during COVID. But there was one thing that kind of was a little bit of a holdup for us. It was so important for us to get that heart rate variability measure, and it was not something that we could not do with having contact with our carers. So consequently, our study was paused for close to 6 months. Now, these are the different questionnaires or scales that we used with our carers, defined by different purposes for which we used them. First, to determine the need, we had those two measures, the resourcefulness scale and the heart rate variability measure. To assess the carer responses, we had measures of perceived stress, depressive thinking patterns that are referred to as depressive cognitions, and negative emotions. And then we had 3 measures of their health outcomes, their physical and mental health, and also a health risk behavior scale, which is actually an indicator for their risk for developing chronic illnesses. A little bit about the interventions. Parallel, all interventions followed a similar process, a 2-part process. First, the content of the intervention, whatever it was presented actually during a single session with an interventionist. And this was a voice-over PowerPoint video that contained the content. For example, for resourcefulness training, we taught the skills that constitute resourcefulness. For the biofeedback training, we taught slow-paced, deep breathing exercises and how to use a biofeedback device that would provide information to the intervention recipient on how well their slow-paced, deep breathing was actually providing relaxation. And even our attention control condition, there was a voice-over PowerPoint during which we described different diversional activities that caregivers could use. The second part was for 4 weeks. It was an implementation phase where the intervention recipient would actually practice the skills or the content, whatever they were taught, whether it was the deep breathing exercises and working with the biofeedback device, or whether it was actually implementing the resourcefulness skills within their caregiving situation, or whether it was using the diversional activities. And during this time, throughout the intervention, an interventionist is in contact with the caregiver. During that 4 weeks, they make weekly phone calls to touch base with how are they doing, how are the skills or the content that they were taught, how is it working, what's not, do you have any questions? And those interventionists are supervised, again, by my intervention supervisor, Dr. Juradovic. So where are we now? Well, we're not done, and I don't have findings to present to you. The study was funded in and around April of 2020, just as COVID was really reaching a peak and we couldn't go anywhere. The study was redesigned, but necessarily paused because we were unable to get the heart rate variability data that we really needed. It was essential to the project. So we needed to work on revamping the data collection timeline. We have in fact, we have completed enrollment about two months ago now. And actually, we exceeded the total target. We have 328, I believe. My team is nodding there. Thank you for helping. Okay, so we have 328 actually enrolled, so we exceeded the target of the 300. And all of the baseline, the first time data, we measure people over at baseline and then 6 months, and then 6 months later. All of the baseline data is mostly collected. We may have one straggler that we're but we're pretty much on target with that. The intervention phase will be completed soon. I anticipate maybe within the next couple months. Final data collection, because it is a study that goes 12 months out, it's going to be probably, I'm thinking the projection is by mid-spring. So, whoops, I'm going back here. So stay tuned. I'll be happy to tell you what the results are. I'm excited to know too. Right now, I'm kind of blinded to the research, as principal investigators typically are with this kind of research. But I want to conclude with some reflections on what the professorship means. So I'm going to switch gears a little bit because I reflected specifically on the three, what I'm going to refer to as the 3 pillars of the professorship: nursing, research, and caregiving, and what each of those mean to me. And so I considered how these 3 pillars really aligned very well with the academic, pedagogical, and leadership model that is represented by the acronym HEART, HEAD, and HANDS. And so for me, applying this holistic model, I view nursing as the work of the heart. And in terms of my research, I see the whole world as my laboratory. And that's where my inquiring mind goes. And I embrace the concept of caregiving to embody the idea that we have 2 hands. We have one hand for helping ourselves, the other hand for helping others. And so I leave you with this final closing thought. In all that we do, may we always have hearts that care, hands that serve, and minds that think within nursing, research, and caregiving, and beyond. Thank you.
Thank you, Dr. Z. I'm just delighted that I can call you Dr. Z now. We have a little ceremony. So you stay right there. Dean Gerson, Dean Musil, Dr. Daenerys, come over. I'm now going to officially give you your chairs and their names, so we have to get the right one for the right person. Dr. Z, stand right there. No. Very serious, very, very complicated set of instructions in order for you to actually be installed in your chair professorship at Case Western Reserve University. Sit down. Congratulations.